Dear students, now you are going to study cloning vectors, Lambda Phage and Cosmid vectors. You have studied already what exactly gene cloning and what are the importance of cloning vectors. So, gene cloning is to get a desired gene or gene product in large quantity. You know, if you want to produce in vitro in large quantity of insulin, you have to take out the human insulin gene and you have to insert into a suitable vector, and this vector, recombinant vector, you have to transfect with the host cell Escherichia coli. Escherichia coli, now it is called recombinant bacterium or Escherichia coli. It multiply in a faster rate. You know that they multiply in a very fast, and they also, along with that, you can see these cloning vectors they are carrying our gene also multiplied to large amount, and we can extract that, or else we can culture these bacteria, recombinant bacteria, in a bioreactor to harvest the insulin in large amount. So, that is the importance of these cloning vectors. So, the gene cloning can be done by two methods, the PCR amplification. So, you know that gene below 5000 base pairs can be amplified in this way, whereas the many genes are bigger than this. So, we have to rely on cell based cloning. So, we need cloning vectors for cell based cloning. Before cloning, DNA fragment must be inserted into this cloning vector. You know, you have what are the essential features of cloning vectors and all you have studied. A cloning vector is a DNA molecule that has an origin of replication and is capable of replicating in a bacterial cell. Whatever this cloning vector, the gene of interest can be easily inserted into that, and it can able to multiply in the host cell or the bacterium to which we are sending this recombinant vector. So, using cloning vectors, the long gene to 20 kilobase pairs to 250 kilobase pairs can be multiplied. Cloning vector is a DNA molecule having a capability to accept foreign DNA fragment and can replicate independently in the host cell. You have studied already plasmid cloning vectors, PBR322 and PUC18 and 19, all this. Today you are going to study the Cosmids and Lambda Phages. First, Lambda Phage. You know, phage is the bacterial infecting viruses. So, you will able to see this is the Escherichia coli bacterium, and it is in now you can see there are just on the plasma membrane so many the bacteriophages or else Lambda phages. So, you will find they are sending their genetic material to inside the bacterial cell for the multiplication. So, these are the Lambda phage it is bacteriophage. Its structure is typical. You have studied the bacteriophage structure. It has got a head, and then there is a tail and the tail fibers, and there is a body. So, you will find Lambda viral genome. So, what is the genome or the DNA packed in the head? It is about 48.5 kilobase pair. It is a linear DNA. It is not circular DNA, it is a linear DNA. So, it is a linear DNA with they have a sticky ends. Sticky ends means you will see these are the double strand DNA. So, this is a double strand DNA. So, this double strand DNA having the single strand sticky ends. You have studied when you cut with the help of Eco R1, it produces sticky ends. Similarly, it has got a 12 bases sticky single strand sticky ends at both the ends. So, it is a linear DNA molecule. What happens, you can see, so these this is linear DNA. When get inserted, it circularize with the help of this. You will find a single strand trail. They are complementary to these sticky ends are complementary. They are cohesive ends. ಬಿಕಾಸ್ ದೇ ಆರ್ ಕಾಂಪ್ಲಿಮೆಂಟ್ರಿ, ದೇ ಆರ್ ಕೊಹಿಸಿವ್ ಎಂಡ್ಸು. ಅದೇನಪ್ಪ ಅಂದರೆ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಸೆಲ್ ಒಳಗಡೆ ಹೋದ್ಮೇಲೆ ಇದು ಲೀನಿಯರ್ DNA, ಇದು ಸರ್ಕ್ಯುಲರೈಸ್ ಆಗುತ್ತೆ. ಇಟ್ಸ್ ಬಿಹೇವ್ಸ್ ಲೈಕ್ ಎ ಪ್ಲಾಸ್ಮಿಡ್. ನಾವು ಬಿಕಾಸ್ it has got a cos site or cohesive ends, it is easily get ligated and it behaves like a plasmid. So that is the advantage. It has got a cos site, cohesive single strand tail. It it hybridize and going to form circular molecule. So this lambda tail fibers absorb these tail fibers absorb to a cell surface, you will find here, and the tail contracts, so ejecting the DNA. So this is how exactly once they get absorbed on the surface, then the DNA of this lambda page will be ejected into the bacterial cell. So the page contains a linear DNA, that's what I said, it's a linear DNA, and it can accept a large foreign DNA and the fragment about 20 to 25 kilobase pairs. Sometimes it can more than 50 kilobase pairs. Different types of page DNA can be engineered, and large size gene of interest can be inserted into this. Hence it is a very ideal vector for long eukaryotic genes. In the host cell, lambda DNA circularizes. Once it is get into the host cell, it get circular organization and can seal the sticky ends. The sticky ends, the cohesive ends, you can see they get sealed, or with the help of the they form a complementary base pair and the ligase will join this. The two ends of lambda DNA bind together forming a circular DNA. So transduction, transfer of host genes from one cell to another with the help of the virus. What we are doing here, you can able to see, this is the lambda page virus. We are taking out the lambda page DNA. It is a linear DNA. Then we are inserting, we are engineering it, that is we are adding essential feature to this long DNA. Then we are inserting our gene of interest. Then after that we are putting this back into this the empty viral cell. So the viral cell, this is now it is free of the disturbing elements which is causing disturbance to the host cell. So it is just having our the lambda page DNA carrying our gene of interest. It has got a, see this is the foreign gene, it is these are the viral DNA, and it is when it is enter into the host cell, it circularizes. Okay, so this is the lambda page you will find. So this lambda page diagram, the lambda page vector diagram, what I have drawn here, so it is having the essential features added, it is engineered. So it has taken from the virus and it has engineered. It has a laxy segment, it has MCS, multiple cloning site, it has a promoter for laxy. So all these genes are added, and it is added with the EcoR1 restriction sites. Then you will find origin of replication site, all these are added. Original the viral DNA part is the cos site, single strand tail. Can you see at 5 prime and 3 prime end, it has a single strand tail, even though this is a double strand DNA molecule added with, engineered with. So for example, we buy cloth and we make the pant out of that, and we add so many pockets, then all these the loops for the inserting the belt and all, no, same thing you can see here. So it is added with the laxy segment, MCS, all these those are required for inserting our gene of interest. So this viral DNA can be engineered and used as cloning vector since 1974. It is not now. Lambda page vector derived from wild type of virus which contains a double strand genome with complementary single strand cohesive or cos site. In the host cell, lambda DNA circularizes because ligase can seal the sticky ends of the cos site and forming a circular DNA. And identification of recombinant page vectors are easy because it is you can see it is easier than plasmid vectors because it has got a laxy segment. So, lambda page vector contains promoter, operator and laxy gene, that is from the E. coli. Hence, this gene inserted to the laxy segment can be easily detected as a colorless plaque you have known, or colorless colonies. It has ORI site and genes for DNA replications. So, what are the advantages of lambda page vectors or applications? Lambda page vectors can easily inserted to E. coli and get replicated. Lambda page vectors contains laxy and it can be easily inserted to the E. coli and can be selected as it's a colorless colonies, you know that. So, it can be in the MCS, the gene of interest can be inserted. So, then after inserting this has to be put into the virus again. This virus will infect the bacterium, sending our recombinant vector into the bacterium. So, once it is getting to the bacterium, it circularizes and it behaves like a cloning vector. So, this will be easily, it can be inserted into the Escherichia coli, and this recombinant Escherichia coli can be selected like a colorless colonies. So, next coming to the Cosmid vector. Cosmid is a hybrid vector. It is the essential features of lambda page vector and the PBR322 will be taken and that is constructed into a Cosmid vector. So, Cosmid stands for cos site of lambda page and the plasmid vector, that is what, it is a hybrid vector produced by recombining useful features of plasmid, that is the PBR322 and lambda page vectors. Cosmid contains ampicillin resistant site, you know, it is taken from the PBR322. Obviously, you will find the selectable marker will be ampicillin resistant gene. Then restriction site, that is the MCS, then ORI site, that is from PBR, then the cos site from lambda page vector. So, lambda page vector ಇಂದ ಒಂದು 250 base pair sequence, so that is concerned with the cos site will be included here. Okay. So, this is from the lambda page and rest of this from the PBR322. So, the lambda page DNA of Cosmid contains 250 base pairs which includes cos site. Cosmid vectors are small, about 2.5 kilo base pair. The total, you know, the DNA, it is about 2.5 kilo base pair and easily packed with lambda head. So, it is this Cosmid, it is behaves like a lambda page, but inserted with the plasmid features. Okay. So, what we can do, it linearizes and we can insert the gene of our interest, then we can pack this in a bacteriophage. This bacteriophage infect the E. coli and sending our you know, the Cosmid into the, it is a linear till it enters into the bacterium. Once it enters into the bacterium, it circularizes into a Cosmid and behaves like a Cosmid vector and behaves like a plasmid. So, Cosmid vector contains essential features of the plasmid vector PBR322 and cos packaging sequence of lambda page. Cosmids, because you know the linear DNA, it is easily get inserted into the, so that is the advantage with respect to the Cosmid and lambda page. So, that we can insert a large size desired gene into this, that is the advantage. It can able to carry 32 to 52 kilo base pairs desired gene or DNA packed into the lambda page part of the Cosmid and easily injected into the host cell. So, this is the linear DNA, can you see the, this is taken from the lambda page and it is engineered and we can you know, insert a desired gene into this, then we can send this into the E. coli, and the E. coli now a recombinant E. coli carrying Cosmid vector carrying our gene of interest. These cosmids packed into the lambda heads can be transfected and replicated easily in the E. coli like plasmids. Before inserting the gene of interest, cosmid has to be linearized. That is why it has to be, it is not, actually it is getting this circular formation once it is get into the E. coli. Till then it will be a linear vector, okay. It has to be linearized, so straightened, that is what, so that it will be have cos sequence, so that the cos sequence, that is the cohesive end, sticky ends. So that is why it is easy to send into the sorry. So what are the applications of the cosmid vector? Cosite allows to pack a large sequence of desired gene about more than 50 kilo base pair. Cosmid vector shows high transformation efficiency. It can easily inserted into the E. coli bacteria and it can be multiplied into large number. Helpful in the construction of gene library. Vectors are small, easily inserted to E. coli. Inside the E. coli, it behaves like a plasmid. Can be cloned into number of copies. Thank you.